Swayam Prabha. Digital India. Educated India.
Hello all, welcome to our course, back to our course on Precision Oncology. Last class we have discussed on the aspects of drug discovery in the Precision Oncology, where we have gone in detail about the master protocols, we went in details about the basket trials, we went into detail about the umbrella trials. Okay, so basically how do this, why is it so many drugs are required? How are these drugs brought under the same umbrella of different arms of treatment? All that we have been totally went in detail in the last session. So now here in this session we will really understand what is exactly the preclinical models used for precision oncology during the drug discovery. So these preclinical models could be even in the cell culture model, the patient-derived explants, patient-derived organoids, and the patient-derived xerografts. For this session, we will be talking in depth about the cell culture, what are different cell lines used, what are the advantages, the disadvantages and the applications, and we will just very lightly touch upon the patient-derived explants and the patient-derived xenografts here. If we really understand, this is a workflow for drug discovery. Cancer is an epidemic disease causing approximately several million loss of life every year around the world. So there is an absence of understanding about the complete cancer biology of each particular tumor. So now could be a key hindrance to the during the progress to for the detection or for the diagnosis or to look for different clinical drugs. So now for that particular to this challenge, initially scientists came up with a model called cell lines. So why do we need these cell lines? Is because to test for the anti-cancer drugs, to test for the efficacy or the therapeutic properties of these particular anti-cancer drugs, we require the cell lines. So they are created in the laboratory to display the key characteristics of the specific cell type, while at the same time most of these cancer cell lines are immortal, because and as a result they allow for the study of a specific cell type without the necessity of returning to the same donor repeatedly. For example, a HeLa cell line, very well established cervical cancer cell line, there are almost around several thousands of studies on this particular cell lines by almost all different cancer groups. If you go across through many of the newbies labs for cancer biologies, yes, HeLa cell line is a starting material for the anti-cancer drugs discovery. So this gives the pipeline for how a cancer for the drug discovery. So the basic research where we have identified or any of the lead compounds, then for that we have the preclinical development where exactly here the cell culture, the patient derived xenografts or the xenografts will come, and then the clinical trials which they are tested on the patients and further go on to your FDA approval so that they can be launched in the into the market. So this represents the systematic representation of the drug discovery process from a basic research which is identified, which is taken care by the target identification, and this will be translated into the market through the approvals of the FDA. So this the good laboratory practices are completely required here for this particular staging. So what exactly are cell lines? Cell lines are the laboratory models, as I mentioned before, and they are taken isolated from an cancer individual, from that particular tissue and from that particular tumor of origin, and they have been propagated over the several years. So there are several repositories to maintain cell lines such as your ADCCs, which maintain the state of the art facilities, so that in order to facilitate researchers with the best cell lines possible, in the sense that the authenticity. So for example, the good laboratory practices are all very well maintained. So and this cell lines are the complete laboratory models, and they are and it is very important if one is working on cell lines to maintain a very good laboratory practices in terms of maintaining the sterility of the areas, or in maintaining good protocols to ensure proper passaging and storage of this particular cell lines, and most importantly they should be maintained contamination free, so that a particular drug tested is reported very well in lines with the standard protocols. So this is the progress and breakthrough of cell lines. So if we can clearly see here from 1907 till 2019 how the cell line research has obtained breakthrough, where here the in vitro cell culture experiments, where they have started the first growth of the embryonic tissues of the frog. So it was established in the beginning of the 20th century, when the first work describing the culture of living tissues. Here it was reported by the Harrison at the anatomical department of the John Hopkins University. And at the 1911, the Rockefeller Institute for Medical Research in New York had T. Burroughs and Alexis Carrel, where they had grown the in vitro culture of tumor tissues derived from the Chicken-Rocket sarcoma. And from then onwards, so they have been, so this particular cell is a very much landmark here, where the HeLa cell line, that is the first human cancer cell line from a cervical cancer patient called Henrietta Lane, was established in 1951. So, and then as it was progressing in the cell line, so they have established that they have identified the mycoplasm, which could be a key contaminant in the cell culture processes. So, and then there is an establishment of the cell line RAGI, the Raji, and which is the first human hemopoietic cell line. So, which is done by the, and this is the Raji is a Burkitt lymphoma cell line, and it is the first human continuous hemopoietic cell line. So, although the Raji cell line was successfully proven to be a model system that generated by EBV virus infection, so the cell line conditions definition was very important to establish its growth, and so that, and this is very one first of the cell kind for the suspension culture. So this is a very again another key important breakthrough or the landmark during the cell line research process. And again here you have the 1970 the K562, which is derived from the chronic myeloid lymphoma. Here 19 and as this research was extensively going on and several groups were adding their own cell lines into the repository, there have been identification of a cross contamination. That is, maybe sometimes a HeLa was not labeled as exactly HeLa or misidentification of cell lines at the worldwide issue. So this has become a very serious issue in the process of drug discovery. So in that time, the National Cancer Institute, which we will be dealing very in detail in this lecture, was started. That is and they have established the NCI anti-cancer screening. Then several in vitro model systems for studying the acute promyelotic leukemia was established, and there have been identifications of many other guidelines and so on. Until now, we have a comprehensive data sets for regarding the genomic, transcriptome and the proteomic profiles for each of these individual cell lines. So this is very important for this particular group for identifying the drug discovery to establish a cell line which is very authenticated is very important. So this NCI screening service has been active from 1990 and it covers 60 different human cell lines. To study the typical cancer models, it is very important to have an intensive understanding of cancer biology in the recent era, And if this particular understanding is totally impaired by, improper models, it is a key hindrance to the research and development. So it is important to understand the invasions, to understand the metastasis, and to understand the tumor microenvironment of this particular disease. So very briefly, this slide shows the typical in vivo models, in vitro models as well as the in vivo models. In the in vitro models, the cell lines and the organoids are very well established. So in this, whereas in the in vivo models, you have the genetically engineered modified organisms, or the PDX models, the zebrafish, and in the later stage the oncopigs, and some of the study groups utilize Drosophila. So this cell lines, they are the in vitro models. They will be representing only the tumor microenvironment. They may not be representative of the complete tumor per se. So they will not be the cell lines. They may have genomic alterations which are characteristic of the original tumor, but because of several passages over the years, they still do not represent the key original tumor characteristics of the original tumor, and they could not have any, because they are the pure homogeneous cell lines. They do not have any of the cell-cell interactions, and they are very much prone to cross contaminations, because in a lab we do not handle only one cell line. When you use many multiple cell lines, there is a possible chance of cross contamination, and it could be costly to culture. But they can go for computational cancer models, where these cell lines, they can be taken up for the genome, the transcriptome, proteome, and the metabolome analysis for identifying specific mutations linked to this particular drug response. So this is a very key important use for application of employing cell lines in the cancer drug discovery. And this will allow, this particular use of cell line models will allow for the identification of potential novel markers, the driver and the passenger mutations, and the driver therapeutic targets, which they can be really taken up further as leads during the drug discovery program. And when we use this PDX models or any of the other models, they require an immunodeficient host. So, and sometimes the tumor, if it's a very active tumor, if it's a very invasive tumor, it may not be able to establish in the PDX models. So, and technically they are challenging to perform. Yes, a little bit of skill set. You need to have a veterinarian to assist in the surgery, for example, any orthotopic models, and they are expensive to model multiple mutations, and the it's a time consuming work, and specific skill set, invasion method for drugs administration and drug absorbance, and to study the endpoints also, there is a lot of animal sacrifice required. So these are the advancements in the cancer models which are currently being applied all over. But however, the tumors from all these particular models can be gone for further computational cancer analysis using any of these four omics technologies. Cancer cell line is an in vitro tumor model that is recorded as an ubiquitous feature for cancer biology, because it shows numerous intrinsic features of cancer, and because it exhibits similar gene expression patterns, a copy number alteration, and a transcription cell profile of cancer cell lines. It's quite similar to the origin of its tumor. So, cell lines are foremost clinical cancer models due to they are very critical, because it's an ease in handling. They are it is slightly relatively inexpensive than compared to the animal models. They are immortal, and they do not have so much cellular heterogeneity, and there is a very fast high replicating rates. So, cancer cell lines, how are they established? They are established by isolating cancer cells from patient-specific organs and then growing them in artificial culture media, which on transplanting again in If these cell lines are put them back into the immunodeficient mice, they lead to the development of the cancer cell line xenografts. Please note, this is different from a patient-derived xenografts, wherein the tumor per se from the patient is implanted into the mice. But here the cell lines are injected in a particular number based on the weight of the mice to get into the mice models. A continuous growing cancer cell line is a very important resource for studying the mechanism of the anti-neoplastic drugs or anti-cancer therapeutics, to study the mechanism of tumorigenesis, and to study the mechanism of any other mechanistic pathways. So there are many several cell lines developed, especially from the American type culture collection. So cancer, as I mentioned before, there is an NCI 60 cancer cell line panel, the breast cancer panel, and the colorectal cell panel. So the primary tumor mutations can be determined by the primary exome sequence. As mentioned before, the HeLa was the first established from a 30-year-old woman, Henrietta Lacks, by the American scientist George Way. So this is a very important landmark as discussed in our previous slide. So what do we do with these cell lines? How do we make them relevant to the cancer drug discovery? So there's something what is called cell viability assays, where the number of healthy cells upon treatment of a drug or a compound is estimated. So this measurement of this viability plays an important role in all forms of cell culture. Sometimes it is the very key purpose for determining a cytotoxic assays, or it can be used to analyze the behavior of the number of cells upon giving a stress condition. So cell-based assays are often used for screening of collection of compounds, as we have seen here. It is used to determine if the test molecules have effect on cell proliferation or show direct cytotoxic effects that eventually lead to cell death. Cell-based assays are widely used for measuring the receptor bindings and a variety of signal transduction events that may involve the expression of genetic reporters, trafficking of cellular components, or monitoring organelle functions. So regardless of the types of cell-based assays used, it is important to know how many viable cells are remaining at the end of a treatment of a particular drug. So there are many varieties of viability assays, as we have shown. So we have importantly, the trypan blue stain assay, the Eoson, the Congo red assays, and where you can measure. So this is a very important, very key basic experiment in cell biology, where the dead cells take up the trypan blue and the live cells don't take up the trypan blue. And again, a very important, an ELISA based method, a colorimetric based method, where we have the MTT, MTAs, and all this particular assays. So this particular MTT assay is nothing but it is based on the reduction of the MTT to formazine, and thereby the formazine is solubilized by DMSO. So the more the MTT reduction happens, the more the viable cells are present. We have again another fluorometric cells, and then the luminometric assays for determining the viability assays. Even a flow cytometric analysis, such as the membrane permeabilities, such as your annexin V binding assays, they are very important to determine the viabilities of cells. So this is a very classic example of how a cell line can be mislabeled or it could be contaminated. Here in this particular experiment, they have discovered that the HeLa cell line, which is derived from a female patient, has expressed a Y chromosome marker. So this cell line authentication is a very major concern in the cancer biology research labs. Mislabeling, replacement of cell lines derived from different tissues, species and individuals, and their contamination with other cell lines is a serious quality control issue which is faced by several of the cancer research groups. For example, the ECV304 is claimed to be spontaneously transformed from human normal endothelial cell line, but it shows that it is a T24 bladder cancer cell. So cross-contamination, mislabeling of cell culture vessels during routine manipulation, and cell line, it's a very common procedure which happens with many of the researchers. So therefore, a cell line verification by short tandem repeat profiling is very important, and this has been assured by many of the top cell line repositories to give the STR profiling upon the delivery of the drugs, a delivery of the cell lines to the particular lab. So, National Cancer Institute team has come up with a screen, a screen, NCI 60 screen, which is led by these three particular important scientists. And since its early introduction in the 1990s, the national NCI panel of 60 cell lines have been used to screen more greater than 1 lakh cancer compounds and greater than 50,000 natural compounds or their extracts for inhibition of tumor cell line growth profile and thereby facilitating going for the FDA approval for this particular cancer drugs. The NCI, this particular, it's nothing to doubt, this particular NCI 60 cell line has been extensively molecularly characterized by the exome sequence, DNA methylation, mRNA, the microRNA expression, protein and studies. The compound profiling data and the molecular characterization of the cell lines, they are all publicly available and they can be downloaded from this particular data website. So, this has served the global cancer community for greater than 20 years, and it was completely operational from 1900s, and it serves for the discovery of anti-cancer drugs. So, this utilizes 60 different human tumor cell lines, which these cell lines represent the leukemia, melanoma, the lung, colon, brain, breast and the kidney cancers. So, this screen is unique in that the complexity of a 60 cell lines. This diagram represents the major development, implementation and use of this NCI 60 cell lines. So, this work was proceeded through 3 phases such as the model development, operation as a primary drug screen and operation as a key utility of the cell line screens. And this work, as I mentioned before, was initiated by Paul and his team. So, there have been several algorithms for comparing results across multiple compounds. Their algorithms, whatever they have identified, they allowed the system to tell or to inform that a compound was unique in its profile of cell killing or cytotoxicity, or it could even suggest the mechanism of action, or it did not share a mechanism of resistance with the establishment reagents. The molecular basis for these 3 distinctive patterns was not first initially elucidated, but the initial linking of the powerful insights came from the compare and the other tools which emerged from the laboratory. That MDR one sensitive compound displayed a common sensitivity pattern that depended upon the presence or absence of the important drug resistant transporter in this particular cell lines. So, the different milestones are given here. So, this is a workflow for NCI 60 screening and secondary testing. So, compound suppliers, they are required to provide information about the biological rationale and chemical structure of the compounds that are to be examined, and duplicates of the previous compounds or representatives of studied chemical classes. If there are complete only analogs, they are all completely rejected. So, this accessed compounds are sent to the central repository and then to testing. They either in the pre-screen model or directly to the US National Cancer Institute 60 anti-cancer, that is called the NCI 60 anti-cancer drug screen. The results are then examined thoroughly by the team of scientists, and the compounds that show a pattern of interest are again sent for rescreening or retesting. The biological evaluation then evaluates this particular screening with reference to the chemical or structures of the compound or any other available literature from the existing information. compounds that have reproducible patterns of interest usually, and those with relatively unique and those which do not. So, they are called, they are the compare negative, are initially tested in the in vivo hollow fiber uh, assays. Compounds and that are further taken down into further xenograft assays as well. So, this is the particular the testing of the drug of interest involves the application of the 2D dimensional. So, please keep in mind this is a 2D tumor cell culture which are grown in monolayers. Testing is carried out on 3 cell lines that are frequently and the most sensitive, for example, the MCF7, the NCIH460 and the SF268. So, and this is the cytotoxicity of the test substance is determined is using the pink anionic dye sulforodamine. So, if the test substance inhibits the growth of at least one cell line, testing proceeds to the next stage comprising. So, this of for the full 60 cell line panel. So, this gives a complete idea of how the drug development group works and how the complete how this NCI serves to use cell lines as a preclinical model during anti-cancer drug discovery. So, this is a systematic overview of the information intensive approach of the cancer pharmacogenomics and the pharmacoproteomics based on the NCI 60 cancer cell lines. Each row of the activity here, A database, represents the pattern of activity. Database A, it represents the pattern of a particular compound across the 60 cell lines. The database can be mapped into a structure S database containing a 2D or a 3D chemical structure descriptors. So, for example, any of the methyl or any of the position in the functional groups or any of the difference in the core structures they of the compound, and the target T database here, and the target T database containing molecular profile information of the cells is completely seen here on the cells. The T database consists of data on the individual molecules, the omics data on the DNA, mRNA, protein and functional levels. The bioinformatic challenge is to understand and analyze this independent study of this independent database separately, and then to integrate each one of these data sets, the information from this data sets to the activity relationships of that particular given drug, and with the public information for any of this pharmacogenomic purposes. So, this is how the inventory of 50%, if there are around 50,000 compounds, 60,000 will less than 1 lakh will come to a 60 cell line screen, and they will go for some of them will go for animal studies and further for their toxicity studies and the clinical studies. So, here we can clearly see that how even though there are so many of this particular NCI 60 has several cell lines. So, but there are some molecular targets for the specific tumor type which this particular cell lines does not address, which we are not. So, some of this target agencies, yes, the status of this target agent in this particular cell lines there, whereas for all this particular important mutations, the important proto-oncogenes, still they are in development, and there are many some targets which there is no that this particular target, it's only for the NCI targets, the cell line targets, only the KRAS and the PI3K, the cell lines, the mutation status is known. So, the cell lines are not very much sufficient for an heterogeneous tumor such as lung cancer. So, what are the different cell culture applications? So, we call this animal cell culture or the cell culture or the animal tissue culture. In current drug discovery programs, it is very mandatory to select cell lines that have been genomically sequenced and they are biologically characterized in terms of identification of the driver mutations or the resistant mutations, so that we know that this particular screen or this cell line will provide an important tool. It will serve as a tool for a good drug design or development program which many scientists are employing. For example, the BRAF inhibitor, that is the vemurafenib, which is very effective against a particular mutation, that is your V600E mutation in melanoma and lung, but it is not, it is ineffective in the same mutation in colon tumors. Thus, in your target specific screening, it is important to characterize the cellular context. For example, where exactly which particular tissue. For example, in this case, the colon or the lung or the thyroid. So, several separate screening methods against this V600E mutant in the colon tumors are now going again, and they have identified overexpressed EGFR or was also an important accessory pathway of tumor proliferation in colorectal cancers, leading to clinical trials of BRAF inhibitors in combination with an anti-EGFR antibody. So, for example, the drug resistant subsets or the drug resistant tumors, there is a need for the drug resistant cell lines as targets for discovery of the second or the third generation inhibitors. For example, your TKI inhibitors. Current screening approaches may have incorporate over 1,000 cell lines and more than 100 varieties of a single histological type of a tumor. So, with the advent of the DNA technology, molecular manipulations has led to the development of several cell lines which can be used as important leads during the drug discovery process. So, this particular diagram gives an application of the cell culture in various uh, technologies. Our relevance is here with the drug discovery. So, we have seen if we have a novel compound that can be identified as a lead compound. So, there should be a lead target in the cell line model. So, can we characterize this particular lead targets, maybe the protein essentially or a key enzyme or a kinase, and they can be targeted, they can be really targeted, or the mechanistic pathways can be elucidated using this drug discovery programs in cell lines. Yes, they are good disease models and toxicity studies before the drug is characterized or given to any other animal model. They have to prove that they are less toxic in a normal human cell lines models. And again, the drugs testing, the personalized therapy, yes, if we can take up this particular cells, maybe the patient derived cells, and you can identify given the same anti-cancer treatment on this particular cell line of the patient X, yes, we have a personalized therapeutics being developed. And no wonder cell lines are also finding very good applications in this food test and nutraceuticals and in regenerative medicine. What are the advantages of using this animal cell culture? Yes, the role of even effect of pH, yes, the temperatures, O2, CO2 concentrations, and can be altered to study their effects on the cell culture. Metabolism, to study cell metabolism and investigate the physiology and biochemistry of cells, they can be really well employed. As I mentioned, cytotoxic assays can be very well studied. The effect of various drugs on the growth patterns of the different cell lines. And these cell lines are very important because they are homogeneous structures. Then a researcher need not be worried if the study can be replicated or not, and they provide valuable biological data from a large scale cell cultures. Specific proteins can be synthesized from a large genetically modified cells. However, they are expensive and it is a very skill set technique that requires complete aseptic conditions. There is required need for trained personnel and the cost of treatment. A cell characteristics differentiation can change after a period of continuous growth in cultures, so which could be leading to entirely different properties from the origin of the host. And there is a low amount of product, maybe whatever the monoclonal antibody or the recombinant protein produced followed by downstream processes, they are all very low. the cell lines are prone to micro contamination via the mycoplasm and the viral infections, and they can be detected, and they are highly contagious. So we have seen how the 2D monolayer structures have cell lines are very important for drug discovery programs. After and we have seen what are the advantages and what are the disadvantages. Yes, they cannot be mimicking a typical tumor environment. So in this are all the it is very important to incorporate a very important tumor microenvironments during the drug discovery programs. So you need to have a non-uniform exposure, which is it is not. Suppose and imagine a 2D layer where the exposure to the oxygen and nutrients is very well same for every cell, whereas in the typical in the in vivo situation there is non-uniform exposure, and there is non-uniform exposure to drugs and compounds. In a 2D layer, in a 2 dimension, each cell will receive the same amount of drug, whereas in an in vivo situation, yes, there is no non-uniform signature here, and there is paracrine signal. Here if we can notice, it is not a single cell which is there. We have a difference of tumor microenvironment combined by stromal cells, then there is a comparison of activated macrophages, the malignant epithelial cells. All of them will try to give a paracrine signals. The cancer cells will give which will help this particular epithelial cells to secrete factors, which will in turn allow for the growth of the cancer cells. And then there is a lot of cell to cell interactions, and there is a gradation. That is, maybe for example, if oxygen is there, if we can really see for oxygen, it is there is a region of hypoxia in the center, but there is a region of high oxygen density in the periphery. So but how do we incorporate such elements into the tumor micro in the drug discovery program? To further that, the many in vitro models have come up where a 2-dimensional structures and then a microfluidic structures. So based on this is the monolayer of cells, and then they had come up with a in vitro model that is like a mixed culture. So can they have 2 cell lines of layers of cell, or and using a Boyden chamber insert where a PET porous, so there is a 2 different cell lines. Here a non-migrating cell line is kept on the top layer, and the migrating cell line is seated on the lower layer. And as such, but the contact is maintained by the exchange of fluids between both. And a 3-dimensional layer sculpture, which is very important, that is here if we can clearly see the cells are forming a monolayer, but here the cells are clustered to form what is called multicellular spheroids. And then you have the mixed cell cultures or your mixed cell culture cellular organoids, and then this spheroids can be done even by a hanging drop. And then we have the different cells which form a tumor microenvironment. They can be placed for during the in a microfluidic device and different devices here. And so a tumor stroma with model based on the co-cultivation of several cells on an extracellular matrix model is completely, it's an organoid based model. This scheme of microfluidic system here, it evaluates the invasive potential of tumor cells, which are the different types of in vitro tumor models are generated. So as we have clearly seen the monoculture, it's a single type, and here we can have a mixed culture, that is, and where there is a different and a Boyden chamber insert, where the two different cell types are separated by an culture insert, and there is exchange of nutrients because they all share the same medium over here. So the non-migrating cells are placed on the top and the migrating cells are placed in the below, and there is a different 3-dimensional cell culture through the formation of multicellular spheroids, if we can see, and the mixed cell culture. Then we have been using schemes of microfluidic devices to develop a tumor microenvironment where channel with cells and hydrogels are given here for the is placed in the central channel, and this So based on the purpose of this experiments, these channels are put with different kinds of medium. And here again the tumor bioprinting models, where a tumor model which is layered along with the layers of stromal cells, as we can see here. Bioprinted spheroids are also there, are also present, consisting of tumor cells, and bioprinted spheroids, which are the models of the tumor models, the tumor stroma, consisting of the tumor cells with the stromal cells. And then finally a glioma model consisting of a 3D printed brain, which is very happening in much. There are several 3D printed organs which they are coming with bioprinted glioma model, and the glioma cells and the macrophages are seen here. So these are all the models which are very actively explored during drug discovery in the cell line models. As I have mentioned, what is a 3D model? We know that in a single 2D model the cells are present as a single monolayer, and here the cell seeding is in a linear pattern and the cells grow in a linear way and they can grow in a single linear way. Whereas if we add a z dimension to the seeding, the cell growth, and where the cells are not allowed to adhere into their surface, and the cell growth where they tend to aggregate, so cancer cells, and then they will form a complete 3D structure. So the spheroid models are very well actively proven to be physiologically relevant than the 2D cultures, because they emulate the key features of the solid tumors. For instance, the growth kinetics of spheroids is similar to the real tumor, where the outer layer, here we can see here, where the outer layer of proliferating cells surrounds the layer of non-proliferating cells, that is the quiescent cells or the dead cells, and as a result there is a necrotic layer in the middle, and that gives an hypoxic condition or the region of low oxygen in the center. Those cells are loosely attached to each other, whereas in the intermediate layer cells packing the ECM are denser. As a consequence, an oxygen density gradient is there due to impaired oxygen function, which is one of the causes of the necrosis in the central region of your spheroid. So this is the main difference, as I was explaining before. So 3D models have shown a great potential. So there is an hypoxic core, and there is a loosely dense of proliferating layer of cells around, and different exposure to drug molecules. So we cannot be treating the same drug concentration as we do for a 2D culture, where we can see that there is a uniform exposure to drug. So this represents, this 3D model that is representative of a typical tumor in the real scenario. So there is a wide characterization of an extensive range of cancer cell lines has offered the researchers for the possibility of developing 3D cell lines. Almost all cancer cell lines can be grown into 3D cultures, and they have been into 3D cultures or 3D spheroids, and they have been exhibit tumor tissue like biological features, and when coupled, when there is a co-culture with the stromal or other immune cells, they recapitulate the in vivo microenvironment, providing to be very much more biological relevant than the 2D counterparts. So this gives a scheme for there are different methods for preparation of multicellular tumor spheroids or the 3D spheroids. So we have the hanging drop method, there is an lipid overlay method, and there are spinner flask and the magnetic cultures. For in all these methods, we can clearly see how the the cells are coming together at the bottom of a hanging drop, or where the surface of the plates, or wherever here the cells are coming up in the liquid overlay method, where we can clearly see the accumulation of the cells here because of a non-adhering adhesive coating to the surface. And then this particular spheroids, they have a typical physiology as mentioned before. They depicts the distribution of proliferating. So this orange cells are distributed in the outside layer, the violet are the quiescent, the middle layer, and the necrotic cells at the violet is here, and these are the necrotic cells, purple at the clue. So the cell density is lower in the outside layer. So this is how a typical and this typically represents the tumor microenvironment, and the spheroids could be. So we have further stepped up now from the cell lines to the spheroids or the MCTS, so that we can imitate a tumor microenvironment to some extent. As very well with the 2D model, yes, the 3D organoid culture models, they are very well applied for tumor cell to understand the tumor cell metabolism and the signaling pathway, and then to understand the invasion and metastasis of tumor cells for the drug delivery system evaluation. Yes, can the drug, can it penetrate, and then it's easy to section this particular model, and then anti-tumor drug screening, very, very important. What are the various approaches used for 3D cell culturing? So we have the hydrogels, where the cells are mixed with a hydrogel matrix and then using a crosslinker, and then we have the 3D bioprints to maintain the scaffolds for recording the 3D spheroids, and then there is something called the microfluidic chips for maintaining the uh, 3D spheroids. So this are all important approach used for 3D cell culture ring. And given the different comparisons, as have been mentioned, this table mentions the different advantages and disadvantages of each 2D or the 3D have over each other. The 2D cultures have a very less physiological relevance, but because of the hypoxic region, the 3D cultures have a better physiological relevance. And in a 2D culture, the culture formation, the cultures are established within few minutes or immediately after few hours after the seeding, whereas it may take few days for the spheroids to form, and it is a challenge to maintain spheroids without much disrupting. So careful technicians, careful methodologies have to be employed not to see the disturbance of the spheroids during the treatment of the drugs. The 2D cultures have high performance, the simple to culture and easy to interpret. There could be variations in the 3D cultures because of complexity of cultures and difficult. So maybe if the same conditions are reproduced every time, it is still the challenge to reproduce the 3D culture drug treatment. The 2D cultures do not mimic tissue environment, but here there is a in vivo condition are replicated. There is no cell or no extracellular environments in the 2D culture, and there is altered morphological characteristics and cell division process. Thus, there is loss of polarity and phenotype. Here some kind of cell division process, the morphological characteristics and diversity, polarity, phenotypic characteristics are retained here. So there is a lot of changes in mRNA, gene splicing, gene expression and cellular biochemistry, whereas they could be all these particular characteristics are representative of the in vitro environment. All homogeneous distribution as mentioned before and unlimited access because of forming a spheroid, the distribution of the drugs or to the access to the nutrients is not as uniform as before. A poor drug metabolism, it could be a drug. It could be the 3D cultures are much more expensive and there could be a specialized skill set. Reproducing a 2D a drug IC50 in 2D cultures is easy, whereas in 3D cultures it is a real challenge. So one more important human tumor histoculture studies, and they were pioneered by Robert Hoffman et al. And they involve the generation of tissue slices from tumor which are obtained from surgical resection, and they were then cultured for extended periods of time using specialized collagen cells. So this ensured that this particular tissues, they have retained the proper stromal component within the TME along with the cancer cells, and this whole thing is retained and it is found to maintain properties of their in vivo state, including the spatial organization and differentiation cell function and the growth of multiple cell types within a single cell was observed. So culture of, as mentioned here, there is this histoculture drug response sensitivity was done. Here where the development for assessing the chemotherapy response. So, for example, you have a patient tissue, the same drug that is being administered to the patient can also be given to your to this particular histoculture or the patient derived explants, and then we can monitor the PD biomarkers in the explant, and then there is an MTT assay for as an endpoint analysis, and then here which we commonly use even everywhere here is the gelatin sponges to support this explant culture. It's just as nothing but the dental sponges where they are inserted in the medium of choice, and along with the supplements or the medium requirements, the tissues are implanted here. So then after few days of allowing the explants to establish, they are treated with the drugs. So here this particular group has used to test targeted therapeutics, yes, and then the PDE correlation with the patient outcomes can also be analyzed here. This is an available preclinical cancer models as mentioned in the first slide of my talk, where there are some models where only the endothelial cells, the cancer associated fibroblasts, the immune cells, the tumor cells, they are all represented in each of these preclinical models. So, for example, the mouse model will try to maintain all this, but along with the cancer associated fibroblasts, and then the explants also will have most of these particular cells and along with the tumor cells. The organoids or the cell lines would be only with the tumor specific, and in all this particular cell lines, so these two, the organoids and the cell lines are they are the in vitro models, whereas the explants, of course the explants are also the in vitro models, but there is the some of them are the ex vivo and the mouse models are the in vivo models. And very interestingly, there are mouse models taking the circulating tumor cells as well. So, this in vitro and the in vivo approaches generally involve deconstruction of the original tumors, and in some cases these original tumors are reconstructed for subsequent assessment of your drug response through the so although the PDX models are the whole motive, they can preserve the integrity of original tumors following immediate transfer. So, the explants, ex vivo approaches such as the patient-derived explant models, they assess that the drug directly in tumor samples obtained from surgery without a deconstruction or a reconstruction. So, the tissue per se, the original tissue, of course, if it has to be histologically characterized or molecular signatures for that tumor have to be obtained. So, this particular scheme shows the cell types available for derivation and use in each model system. So, this represents the three clinical models here again, where we have the patient derived xenografts. For example, the tumor from a lung or from the prostate or from the breast is implanted into the mice for the first generation xenografts, and once this is established, they are treated with the same drug as the patient goes through. So, they can be used as the tumor avatars, the mice models, and then after further expansions into your F0, F1 and the F2 generations, the efficacy of the drug will be measured by the decrease in size of the tumor. So, the tumor volume. So, upon treatment and how the vehicle control in the absence of a drug, how the tumor volume increases, and upon treatment how the tumor volume decreases is clearly shown here. Similarly, in the organoids or with the single cancer cells, that is your patient derived cells. So, this particular effect of these drugs can be very well studied. So, here this gives a typical model of the patient derived organoid model, where the scientists from the 20th century, as I mentioned before, they began to study the 3D culture of cancer cells, and then but 3D culture was not fully representative or widely because of this, or recognized or widely applied because of this technical constraints, as I mentioned before. So, they have shown that it's the patient derived tumor tissue is firstly digested into single cells and transplanted into a basement membrane exactly with the growth cells, that and would be reorganized successfully in several weeks. So, the idea of the organoid cultural has been very well established for several millions of time, and several important publications have come in picture. So, patient derived organisms they have the advantages and gene editing, immunology research and high throughput than PDX. It is, and it can be having a lot of high throughput because it's only a 1 mm dish or 1 mm culture tumor tissue is enough for the organoids to be established. So towards the end of 20th century, many scientists have started from the 3D culture, and they are very well progressing towards organoids, and which are very important for in their drug discovery processes because of they represent the whole tumor. Patient-derived organoids, as mentioned before, they show structural features with the primary tumor of origin. They are such as the expression patterns, the copy number aberrations, the transcriptome landscape, and the mutation status of the tumor is completely retained in this particular patient-derived organoids or the explants. So, after the surgically resected tissue, it is cut into 4 pieces, and then the original tissue along with this explant, there is a confirmation of similarity between has to be done between the tissue and the explant. So it is done by a histology or the the HAD or the head to staining, and by sequencing, copy number alterations and mutations screening. After that, the drug screening is then performed on the explants or the organoids to check for the effectiveness of the drug, or to check if the patient will definitely respond to that particular drug. Here we have to first keep in mind the drug is directly being given treated onto the tumor, whereas in the real scenario the drugs are administered intravenously, and there is another passage route for the tumor to be accessed by the drug. So, a drug screening is then performed to see the results can then assist for the clinician for what treatment can to be used for the patient. So, there are different methods. The development of organoids as an ex vivo model system has revolutionized the primary and clinical cancer studies during the last decades. The PDEs or the organoids are infinitesimal they are representative of human organs and tissues, and functional features and structures of the selected organisms are very well retained or represented. So, the patient-derived organoid cancer model is developed by tumor cells isolated from the tissue of cancer patient and there is the complete extracellular matrix of the specific culture media, which can develop into a cancer organoids. So, organoids are also amendable for further molecular cellular characterization and by manipulation of several genetic tools, and that can help in causing further approach or identifying the causative approaches for cancer in etiology. So now we have just introduced the term patient derived xenografts, where we are using either immunodeficient mouse models for establishing the tumors directly from the patient. So this mouse models, immunodeficient mice models, will harbor the tumors. And the drugs can be tested on this particular mice. So in 1969, Riegard and Povelson successfully transplanted the human colonogenic tumor tissue into nude mice. They have done it in University of Copenhagen, and they have established the first PDX. But however, till 1990, because of the NCI 60, as I mentioned before, they did not promote the use of mouse models because of the because of harming of injury to the animals, or to minimize the usage of animals. The NCS 60, because of it was so widely employed and widespread, its applications were all over. So the PDX models were not coming up into line. So, but however, NOD skid mice was first developed to establish the PDX model in Johns Hopkins University, which greatly promoted the development of PDX. So this was the first PDX model. So again the PDX model gained popularity at 2006, during 2006, at the beginning of 21st century, where there are many increasing number of institutes, especially the pharmaceutical companies, prefer to choose the PDX models in all their pharmacodynamic studies. In 2014, the PDX was used in the science model, was shown in the science journal model to human body. So it was elected as a covers to cover, and this is when the NCI overhauled the NCI 60, this 60 cell lines in 2016. So they have declared that this particular cell line models will be retired in this particular year. So so now of all, many of the pharma companies, many of all the research labs and the research institutes are racing towards to develop PDX models, and only this is the perfect model for evaluation of the anti-cancer drug treatment. This mice models are very key important models, because they represent the whole tumor, and the toxicity or the effect of this particular anti-cancer drug on the particular mice with that particular tumor type is very personalized, or it's very patient specific. So a patient may be harboring any particular mutation, and for example, and this particular drug does not respond to that, or and the tumor does not respond to that particular drug. Yes, this patient may not be taking up this drug, and the drug treatment or the targeted therapy can be circumvented for that particular patient, so as to avoid unnecessary administration of drugs. Here, this is the patient derived models in cancer research. As mentioned, they have been for very fundamental for use of novel new targets, and for the studying of the mechanistic properties, and then pre-experimental of all this particular clinical trials, and in the clinical, as I mentioned here, personalized treatment. Yes, a patient specific treatment models can be coming up. For example, for all the clinical trials, especially in the CAR-T trials, this PDX models are very important, and these drugs can be evaluated for their cytotoxicity in all this PDE and the PDX models, and the mechanism of pharmacodynamics can be well studied here. So in this whole in this session, we have come to the end, where we have totally in detail the applications of cell culture during drug discovery in precision medicine. So we have totally in detailed about the different examples of cell lines, what are the different advantages and disadvantages of using cell culture, and how can they be easily be manipulated, mislabeled, or they could have, the cell lines models could have lost the original characteristics of the original tumor. We have totally in detail seen the 3D culture. We have seen how the 3D spheroids, what are the different scaffolds, the hydrogels, we have the microfluidics to establish 3D cultures. And then we have totally in depth and seen how the patient derived explants can be established, and how they are very much good avatars of the tumors, and the same drug what has been administered to the patient can be tested on this patient derived explants. And then we have seen the very in brief about the patient derived xenograft models, about the mouse models, how the same tumor can be propagated in mice along with the administration of the inhibitors. So this brings to the end of our session. Thank you.